Good afternoon. I'm Mary Kaldunski from the Rat Genome Database. My talk is about enhanced hybrid rat diversity panel resources at the Rat Genome Database. The hybrid rat diversity panel has been talked about in other talks. It's a broad selection of classical inbred strains and 2 recombinant inbred panels, which are designed to maximize genetic and phenotypic diversity, which also maximizes the power to detect and fine map genetic loci associated with complex traits. At MCW, HRDP is a funded program to rederive, sequence, maintain, and especially distribute HRDP strains. And we've included the HS founder strains. Yay! The HRDP information is organized on a portal page, which is accessible from the RGD homepage through either the Phenotypes and Models drop-down menu or the box on the front page. The portal page provides a description of the construction and format of the panel, and everybody uses Dr. Saba's beautiful picture. The original strains are listed on the portal page. You can't probably see them, but a number of them are noted with an asterisk, and those are the ones that have been sequenced to date and loaded to the variant visualizer tool. Almost twice as many strains have been sequenced, and they will be loaded to variant visualizer very soon. IT promised. VCF files are available for download for all of the sequenced strains, whether they're loaded to variant visualizer or not. Each strain has an RGD ID that is a link to a strain report page. Shurjen showed some of these pages earlier in her talk. They're compiled information of all the annotations, references, QTLs, provenance of the strain, and further links to RGD tools like Variant Visualizer and PhenoMiner. Some of the information available for RGD, because we like numbers, 228 curated references with 101 qualitative disease annotations and 226 mammalian phenotype annotations. There are over 100 studies curated in PhenoMiner, which include over 10,000 quantitative phenotype records and 14 or so million variants loaded in Variant Visualizer for the first 41 strains, and more to come. These numbers are for exact HRDP strain IDs. They do not include closely related strains, substrains, or strains derived from HRDP strains, or their numbers would be higher. The genetic similarity between the HS stock founder strains and the HRDP strain that is the best match has been quantified by Dr. Palmer and colleagues. And on that original HRDP portal page, those strains are noted with a double dagger symbol. The HRDP portal page at RGD provides an email contact link. There in the corner. Yeah, there it is. To HRDP investigators for discussions of strains available for distribution. Embedded in the text are links to the Variant Visualizer and PhenoMiner tools for HRDP specific data. The Variant Visualizer link will open with the 41 loaded strains pre-selected. Enter a gene ID and click Continue to generate a variant display. The Variant Visualizer output report is on the left, and on the left side is a list of strains, such that each row in the output is a strain, and the columns are positions across the chromosome. In this case, it's the gene GMAP5 on chromosome 4. Selecting a particular variant will open a variant details page. That has all the transcript information, the assembly, the position, intron, exon, and polyphen predictions for benign or possibly damaging. This is just a reminder that the other option on the HRDP portal page was PhenoMiner. PhenoMiner will open with all the available HRDP strains pre-selected and all available phenotype data for those strains in a downloadable report table. Last time I looked, there were over 226 records. The graph can't display at this point because the phenotypes have multiple units of measure. The filters on the left side are available to make selections. For example, if you pick the first trait in the list, that limits the number of phenotypes to a single unit of measure. In this case, it's vasodilation percent in response to hypoxia. Alternatively, you could pick the Edit Query button, which is at the top of the page. The Edit Query button will return you to the data selection page for PhenoMiner. From here, you can completely tailor the list of strains, clinical measurements, any other parameters. The Generate Report button will return the user to the data interface page with the table. As Shurjen pointed out, on the graph display, the bars are composite data input by curators from the literature. The red dots are individual rat data, mostly submitted by investigators. And both data types are downloadable, as she also pointed out. Hovering over a bar or a data point will pop up a display of all of the parameters for that particular data point. Those are all the pieces of information that are stored for every data point. For a second, I'd like to revisit the issue of closely related strains and substrains. In some cases, very little data was found for the HRDP original strain. Curators spent the better part of a year searching the literature for phenotype, quantitative phenotype data for the HRDP and HS founder inbred strains. And in some cases, we didn't find much. More data can be found at RGD by including closely related or derived substrains. For example, I'm going to use the strain search tool. One of my favorite strains is Lu MNCR. That's an HRDP strain, because that was my assignment for go find data for this strain. And that strain was an HRDP original strain, which was given to NIH and then transferred to the RRRC for embryo cryopreservation. And the RRRC is where we're trying to rederive the HRDP strains. So if you put, yep.
5 minutes.
Oh, not a problem. Searching for Lu M, you can see that there are more strains available than just the Lu MNCR. There's the Lu BBB, which started as a gift from Dr. Bazin, who first derived the Lu/M in Louvain, Belgium. The Lu M WSL strain is still in Belgium and is derived from his original strain. All of this information, the provenance of the strains, the relationships, their phenotypes, their annotations, are all available on the strain search page, the strain report page, this thing. If you return to PhenoMiner through either the little blue PhenoMiner icon boxes or the report page, you will return to the data selection page and you can input whatever strains you want. And from there, we can see for the Lu/M, which is the original Dr. Bazin's strain in Belgium, there's 67 phenominer data points. And for the one in Berlin, there's 12. For the one still in Belgium, there's 136. Where for the Lu MNCR HRDP strain, there's only one data point in all of RGD. And that's for the disease plasmacytoma, which is what the strain was developed for. In summary, HRDP has a wealth of data available at RGD. The portal page under phenotypes and models holds the strains and links to the strain report pages. The strain report pages have curated and imported data and links to the tools Variant Visualizer and Phenominer. I would like to acknowledge our teams, the Rat Genome Database, and the Hybrid Rat Diversity Program at MCW, and our funding, and everyone who uses RGD. Thank you.